Our next presenter is Dr. Aysha Akhtar, and she has a bit of a different angle, which I think is very important and needs to be taken into account when we talk about this issue of animal testing. So Aysha Akhtar, MD, MPH, is the president and CEO of the Center for Contemporary Sciences, which is pioneering the transition to replace the use of animals in experimentation with effective human-based technologies. She's double board certified as a neurologist and preventive medicine specialist, and she has a background in public health. She has served as the deputy director of the U.S. Army Traumatic Brain Injury Program, developing the Army's brain injury prevention and treatment strategies for soldiers. And as a commander in the U.S. Public Health Service Commissioned Corps, Dr. Akhtar frequently deployed to assist with national public health emergencies. For a decade, Aysha was a medical officer at the FDA, most recently in the Office of Counterterrorism and Emerging Threats, implementing studies on vaccine effectiveness and safety, and using her security clearance to develop national preparedness strategies for public health threats. She's a fellow of the Oxford Center for Animal Ethics, the author of 2 books, Our Symphony with Animals on Health, Empathy, and Our Shared Destinies, and Animals and Public Health, which argues for the need for health institutions to include animals as part of the public in public health. Look for her in the TEDx speaker series, because she's also given TEDx talks. I'm going to turn it over to Aysha to reflect on the next presentation, the next set of issues there. Thank you.
This is a picture of my dad, my Abu. And first of all, I just want to say I am from Pakistani descent, and we are not vegans by culture, although my family and I became vegans 30 years ago, which was definitely a rarity among Pakistanis. But this is my Abu, and my dad has had the diabetes. He suffered from diabetes for many decades. And as a result, he frequently experiences a type of pain called neuropathy. It's a type of nerve pain that, if any of you have ever experienced it, it's a very uncomfortable, it's a very different kind of pain from the pain you might get if you cut yourself or, accidentally burn yourself on the stove or something like that. It's truly a miserable pain to have. And there's really has been nothing that has helped his pain. And I've seen him suffer again and again through this neuropathy when it flares up, with nothing really that we can offer him that will take care of the pain. And as JAINA also mentioned, I was working in the Army on their traumatic brain injury program. And for soldiers, traumatic brain injury is a big deal. It's a huge deal. It's a big problem. And really, we have nothing to offer soldiers. There's no treatment for head injury. The only treatment we have is what we call supportive care, meaning we make sure they're properly nourished. We take care of maybe symptoms that result from head injury to some degree, but we have no treatment for the head injury itself. We have nothing for that, despite the fact that our military and military throughout the world have poured billions of dollars into bashing animals' heads. No exaggeration, bashing animals' heads. The National Football League has done the same thing, because football players and head injuries is a big thing. Billions of dollars bashing animals' heads. We have nothing to offer these people who have head injuries. And then, of course, when I was at the Food and Drug Administration, I saw treatment after treatment that was lauded, that the community thought this was going to be the next big thing. Oh, there's so much hope. This is what's going to, be the treatment that's going to really help for HIV, or really help for stroke, or really help for this type of cancer, and so on, only to see them fail when they are tried in humans. So my experience here has really caused me to reframe the ethics. So to not just ask, is animal experimentation ethical for animals, which of course it definitely is not, but also, is it even ethical for us? Is it ethical for humans? And to answer that question, we need to ask, is it an effective use of our resources? And does animal experimentation reliably inform human health? Now, what I'm giving you is an abbreviated presentation to fit into the time limit we have here. But online and on our website, there are longer versions of this presentation if any of you are interested in learning more. But so what I'm going to just talk about is, so Alka and Shalene talked about a lot of different kinds of experimentation. There's a lot of academic work, which is called basic research on animals, and that's where most animals are actually used. And there doesn't need to be a direct clinical benefit when you're talking about academic research. So you don't, as a researcher, you don't need to justify that type of experiment on animals with some kind of direct clinical benefit to humans. But if we just looked at one type of applied research, and when I talk about applied research, I'm talking about the areas of research where there's supposed to be some kind of direct human benefit. So this is really where we're looking at drug and vaccine development and at chemical testing to before they're used on the market, right? Chemical testing for toxicity. If we just look at drug and vaccine development, it's been noted more than about 15 years ago by the Food and Drug Administration, by other heads of some of our public health organizations, that overall, there's a 90 to 92% failure rate in drug development and testing, meaning that between, actually it's closer to 90 to 95 now, 90 to 95% of the vaccines and drugs that proceed to human studies actually fail. Now think about that, a 95% failure rate. If you were to hop on a plane and the pilot said you had less than 5% chance of landing safely at your destination, I guaranteed everyone will hop back off. I mean, think about this. This is about a 5 to 10% success rate. In what other industry in the world do they accept that kind of dismal success rate or that dismal failure rate? But for some reason, it's accepted when it comes to the drugs and vaccines that we put into our own bodies. So there were many reasons offered for why we have such a high failure rate in drug and vaccine development. And one is that there are potential higher regulatory hurdles, meaning that the Food and Drug Administration and other types of regulatory agencies that oversee these kinds of testing throughout the world have high hurdles, high bars to meet, which is definitely a causal factor. Another offer proposed is that clinical trials, and here we're talking about human trials, are very long and expensive to conduct, which they definitely are. They're difficult to conduct. You have trouble sometimes recruiting enough patients. Sometimes patients drop out. Many times patients drop out before the studies are conducted. They're expensive and they can take years to conduct. And so sometimes drug companies may just run out of money during this process. They go bankrupt in this process, or it turns out to be, too expensive for them to continue, or they're not able to recruit enough patients into the clinical trials, or too many drop out. So that's a barrier for them. But one of the main significant factors that has been shown again and again to be a high, and I would argue a major, if not the major cause for why we see such a high failure rate in drug and vaccine development is the poor predictability of animal experiments. So, and Alka brought this up earlier, with rare exception, I'm talking about in the single digits, the Food and Drug Administration will always ask for every new drug and vaccine that comes to them when a pharmaceutical company brings what they call a new drug application or a biological application for vaccines. They have to have been tested on at least 2 different species. Usually one species is a mouse or a rat. The other is going to be a cat, a dog, usually dogs, pigs, and monkeys. So what this means is that those drugs that are allowed to proceed to human trials have all been tested on animals for safety and effectiveness. So all of this, what this all means is that 90 to 95% of the drugs and vaccines that have been found, that have passed animal tests, that have been found to be safe and effective in animals, fail when tried in humans. And most of them fail because not because of long, expensive clinical trials or because of higher regulatory hurdles. Most of them fail because they are found to be unsafe or ineffective in humans. So think about that. Such dismal, poor predictability of animal experiments to tell us whether a drug or vaccine is going to be safe and effective in humans. This was a study. There was a back in 2007. Now you would think, we've been doing animal experiments for decades, for centuries, and you would think there's a valid reason, right? The scientists have been doing it because they've studied it and they have found that it's effective. They have proven that it's effective, that it's reliable, that it's predictive. You would think that this is what science is supposed to be about. But actually, no one really thought to ask whether animal experiments are really reliable and really informative of human health until relatively recently. And so one of the big studies that really asked this was a study that came out in 2007. This was a study published in the British Medical Journal. And these researchers, what they did is they wanted to see how well do the animal studies match what we see in humans. And so what they did is that they looked at six different treatments for 5 completely different kinds of health problems. The health problems include respiratory distress syndrome, head injury, osteoporosis, hemorrhage, and so on. And what the researchers did is that they gathered all the animal data on these different treatments and all the human data on these different treatments, and then they matched the results. They wanted to see how well did they actually match. And what they found was that the animal data only matched the human data 50% of the time, meaning that the animal experiments were no more likely to predict whether those treatments and vaccines were going to be safe and effective in humans than a toss of the coin. Now, this is one of the first to come out that did this, and this is what's called a systematic review. It's the highest form of scientific evidence in the biomedical research world. And then since then, there's been multiple studies that have come out that have shown similar results again and again. So let me give you just a couple of examples of just two clinical examples of where animal experimentation has failed. So if we look at stroke experiments, there have been more than 700 treatments that have been found effective in animal experiments in stroke. Of those 700 treatments, about 150 moved on to human clinical trials, and not one has proven effective in humans. Not one has proven effective in humans. If we look at HIV vaccines, for decades, scientists have been trying to create HIV vaccines to prevent HIV. This includes Dr. Fauci, who is so lauded, but he runs a primate laboratory, and they do a lot of HIV vaccine testing and other types of vaccine testing on animals. Most of these vaccines were tested on chimpanzees until relatively recently, when chimpanzees were mostly phased out in experiments, and they are still conducted in non-human primates. So our closest relatives, in a sense. So more than 85, and that number is actually higher now, more than 85 vaccines to prevent HIV have been proven effective in animal experiments. Of those, more than 30 have been tested in humans, and not one has proven effective in humans. Just in last year, another HIV vaccine that was 10 years in the making, billions of dollars went into it globally, worldwide, and again, that one failed. And as a matter of fact, some of the vaccines have actually, when they were tried in humans, actually suggest that they increase the risk of HIV in humans. So not only did they not work, they might have been worse, that they increased the risk of HIV in those people tested. These are just 2 examples, and we can go down again and again and again, disease category, disease category, disease category. This is a kind of an overview of the drug development pipeline. And there's a couple of things I want to point out. First of all, so what it shows is that in the initial phase in drug development, a pharmaceutical company might be looking at 10,000 compounds to see if there's a potential that they could be therapeutic for a certain disease. And after that initial review, about 250 may move on to what's called preclinical studies. And when we're talking about preclinical, we're talking about before human trials. So this means in vitro studies, computer modeling, and animal testing. And I will say most of this, the main decisions are based on animal testing. In this phase, the main decisions whether or not to move a drug forward is based on animal testing. Then you move into clinical trials, that's human studies, and then move into whether it's approved by the Food and Drug Administration in the US. So on average, it takes about 14 years for a drug to go through this process. For vaccines, it's longer. On average, it takes 17 years for a vaccine to go through this process. I do want to point out that there's this image suggests that 1 out of 5 compounds that go through human trials make it to market, which is not true. We talked about that. It's actually less than 1 out of every 10. So that is not quite accurate. But the main thing I wanted to point out is between the preclinical and the clinical trial phase. So let's say about 250 compounds are tested in the preclinical phase. Out of those, only about 5 move on to humans. What happened to the 245 that did not get tried in humans? How many of those did not get passed on to humans because they didn't work in the animals that were tested? But how many of those might have worked actually really well in the humans, but were never given the chance? So one of the things we have to worry about when we think about animal testing is not just how many failures there are in whether something works in animals and then it doesn't work in humans, but the other way around. How many didn't work in animals but could have worked in humans? How many did not work in those species that were tested but may have worked wonderfully in humans? How many missed opportunities has occurred? So to summarize, the human harms that can result from a very ineffective animal experimental system, which is really the crux of so much of our biomedical research, is that often people who enroll in clinical trials suffer adverse events that were not predicted by the animal tests. Maybe that's happening with some of the vaccines with the blood clots now, I don't know. But many times we've seen about 30% of the drugs that do not pass human trials are because there are safety problems that are found in those human trials. Safety problems that were either not detected at all in the animals or not detected enough, or not seen as a high risk in the animals tested, but they were a high risk, obviously enough in the humans to abandon those drugs. There's also wastage of time, money, and resources, as I talked about. And then there are missed opportunities. How many drugs did we abandon? How many vaccines were abandoned that would have just worked wonderfully, that could have maybe been significant effective treatments for head injuries, that may have been something that really would have helped my dad's pain? How many of them did we abandon and pharmaceutical agents abandon because of misleading studies in animals? So if we don't do animal tests, what do we do? And so there's actually been a revolution in biomedical science that's really occurred over the past 10 to 15 years. And I would argue that this revolution actually would have occurred sooner if more of our funding resources were directed into this line of work. But the revolution that's really taking place is in creating testing methods that are based on human biology. And when you think about it, it just makes sense. Why are we studying the biology of a mouse? Why are we studying the biology of a rat or a cat or a dog or a monkey if our intention is to try to understand human biology, human diseases, and find treatments for human illnesses? It doesn't make sense to be studying other species, because no matter what role animals may have played in the past in animal experimentation. So like if you look at when animal experiments first started, people were cutting open animals, looking at their circulatory system, and there are obviously similarities in the cardiovascular system of a cat and a rat and a human. But we've now moved far beyond that in biomedicine, and we are looking at the subtle nuances of molecular biology and genetics. And here, the differences between species can have a profound impact. So it's important that we get back to studying human biology. So we have now very different techniques now that are coming much more to the fore. One of the big revolutions over the past 20 years is what's called a human on a chip. And these are methods in which an organ, a human organ, is distilled onto a microchip, which is where everything happens, where disease takes place. It's at the micro level. When a drug works, it's at the micro level. If there's going to be adverse events, it's going to be at the micro level. And so you basically recreate a, a 3D, the 3D architecture of a full lung as much as possible. And it mimics a full organ in structure and function to a high degree. And so these organs on a chip have actually been used now much more in the development of these COVID vaccines because of the reasons that Alka mentioned earlier, because there's a need to really use the best, most effective, and fastest testing methods possible to check whether a drug or vaccine is going to be working against COVID. There are other types, and so what we have now is actually there have been multiple organs of chips created: a kidney on a chip, heart on a chip, lung on a chip, gut on a chip, even a mini human brain on a chip, and now they're being connected through lymphatic systems, circulatory systems, and eventually through a nervous system to actually create the human body on a chip. So you can actually see how something works throughout the entire body. There's also called organoids. These are actually miniature versions of organs. These are actually being 3D printed through those bioprinters now. And again, these are human tissues. These are human organs that you can use to study in the lab. There's things called virtual humans. There's a wealth of clinical data, human data out there that is not being used to the max by any means, that you can use to tailor a drug, to predict whether the drug is going to work against a certain marker in the human body, and so on. The Human Immunology Project is a type of project that combines multiple methods: pathology results, autopsy results, clinical trials, in vitro studies, and imaging studies. And those are some of the others that I mentioned here. Now, a couple of things I want to point out is that no one of these methods is by any means perfect. There's a lot of difficulties, as you can imagine, in being faithful to trying to recreate something here on a chip, for example, that's faithful to the human body. And no one of these methods is going to give us all the answers we need. No one is going to give us all the answers we need. The thing to point out, though, is that there has been so little funding into these areas in comparison to funding that goes towards animal testing. And so these methods can continually and will continually be improved upon and become more faithful to human biology and the full human system if we can put more funding in that area. And again, it's going to take a combination of these and other methods to really give us the best, most accurate picture of what's happening in the human body. So just a quick word about our organization, the Center for Contemporary Sciences. We just launched in May of last year, just as the pandemic was starting. Yay, lucky us. Our bad luck to be launching during a pandemic. But our vision mission is really to save and improve lives, all lives, because the work that we're trying to do is a win-win for humans and animals. The more we can move biomedical science towards using human-specific medical research methods, the better it is for animals, without a doubt, but also the better it is for humans, because these can be far more effective and superior testing methods. And so I will, and basically just an overview. I don't know if I give that. No, I didn't. So we trying to work with the scientific community, trying to drive investment into these technologies. We're going to work eventually on policy to try to force NIH to put more prioritization of their funding into these types of testing methods, educating scientists, educating the next generation of scientists in these testing methods, so they come out of their studies not getting ready to do animal experiments, but getting ready to do research using these lovely techniques, and so on. And that's just a brief overview of what we're trying to do. So I'll stop there. There's my contact information if you want to reach me. That's our website. And most importantly of all, that is my Nessie and her soccer ball, one of the loves of my life.